Other is in the vet school, so he'll be back again, I'm sure, at some point. But he does a lot of research in cognitive neuroscience and memory, so if you have any advice and expertise you can present to us is most welcome. So take it away.
Thanks, Randy.
I'm going to sit at the back so I'm not blocking you.
Thanks everybody for coming out. I'm real excited to have the pleasure and honor of speaking with you guys today and share with you a little bit about some of the research I've been doing that looks at learning, memory, and the role of the hippocampus in memory. Last time I was here, I think I spoke a little bit more about some data that I had about non-conscious memory, and today I'm going to pivot and instead talk more about brain states that we've identified, both cognitively and electrophysiologically, that seem to be related to cognitive performance, and particularly talk about some ways that we found that some aspects of cognition can be enhanced, but also identify some of the things that we've identified that seem to be associated with some deleterious effects to cognition as well. So basically the general gist of what I'm hoping to cover is we'll talk a little bit about learning and memory and the role that curiosity plays. I'll talk some more about some brain states related to memory success, and then how some approaches that we've taken so far have identified some ways that might be very amenable targets for enhancing cognition, enhancing performance. And then I'll try to finish off a little bit at the end if anybody's interested in what I call some evidence-based sort of medicine or sort of prescriptions for ways that we can improve our memory a little bit and things like that. Randy mentioned that there might be some people with interest in education and learning and stuff like that. So I tried to add a little bit of that flavor at the end for some of you guys who might be really experienced memory theorists, or some of that might be revisiting some things that you might be familiar with, but we'll see if we have time to capture some of that. As I start, I want to introduce you to someone who's very near and dear to my heart, and hopefully give a scope for the type of memory that I find interesting and that I'll be focusing a lot about today. So please meet Juno. She's about 1.5 year old greyhound puppy. She's very near and dear to our heart. Just like most dogs, she likes to play, goof around. This is her actually in Lansipine, run around on some trails in the back over by Mount Harmon. She likes to swim, long runs on the beach, all normal, very normal puppy things. She likes to travel. She loves the open road. When she was a young puppy, she liked riding shotgun. But as she's grown a little older, she's gotten a little big in her britches, and now she likes to drive when she has the opportunity. As you can tell, this is from America because the wheel's on the left-hand side. But what I would like to do is suggest that you might think for just a moment, if you only had memory for those sentences, but without the rich contextual information of those colorful pictures that otherwise weave that nice fabric of what we call episodic memory, our ability to mentally travel back in time and retrieve the episodes that we experienced before, as distinguished from just the semantic information such as Gina likes to play, she likes to swim, she likes riding shotgun, prefers to drive. They're just semantic pieces of information. And when people have clinical disorders of amnesia, or sometimes if we just have forgetfulness of certain things, sometimes we lose that ability to have these rich, contextual, colorful aspects that I would submit really provide the backbone of what we anecdotally refer to as memory most of the time. So this type of episodic memory is something that I focus a lot on in the research, and that I'll talk about in this particular aspect. Because the idea is that our semantic memories do relatively little to enrich our lives in the same way that we can weave these rich tapestries of our episodic memories to provide us with that more cohesive ability to do what Edmund Tolding referred to as mentally travel back in time and re-experience something that was special to us. Or perhaps that was just mundane, but yet we need to retrieve for something awful like an exam or a test or something like that. Okay? So I'll talk a lot about that, and basically one of the central questions that my research is interested in is how do these episodic memories operate, and sometimes how do such episodic memories fail. So I'll talk a little bit about that, and as I noted, I'll try to address some questions about how we can improve our memory performance, sometimes prevent our decline, and focus in about what we've identified, essentially accidentally by trying to look at some of these episodic memories, which is the idea that there might be some brain states that could be related to supporting our abilities to mentally travel back in time, retrieve these rich, contextual, detailed episodic memories, and ultimately, perhaps, find ways that we can enhance that ability to engage in our cognition. So one of the themes that I'll try to revisit as we move along, and that sort of drives my research, is what if we've been looking at a three-dimensional picture, but with only a two-dimensional lens? So what I mean by that is multifold. One, you can think of it just in terms of methodology, which is oftentimes we might look at experimental questions with one, sometimes two methods convergent if we're lucky, but occasionally we're able to integrate additional forms of methods. So I do some work with clinical neuropsychological patients. We measure behavior and then we'll record EEG at the same time. And what we've been able to do is identify some times where when we converge those three methods, we can gain greater insight than times when we can identify things with those other two-dimensional approaches. And likewise, more conceptually, the idea that perhaps we've been looking at things with a two-dimensional lens in our broader conceptual framework of how we understand memory and cognition, which is in basic response to a stimulus. And I'll talk a little bit more about that as we move on. And suggest that perhaps if we expand our framework to look at the types of activity in the brain states that might be happening before that stimulus-response relationship, we might be able to gain some insight into what might be happening through that, what I call that third dimension, that has traditionally been neglected otherwise. So, that's sort of captured in this far side picture, our two little amoeba, right? Sort of representing our normal scientific approach across the pantheon of science for the most part of the modern era of memory and neuroscience, which is stimulus, response. Don't you ever think, right? So you have the one amoeba talking to the other, just watching the television. And so what I'd suggest is that this part here, don't you ever think, there's got to be more to cognition than just stimulus-response relationships. And that's kind of the essence of what I'll be trying to tap into in today's conversation. And so I'll sort of embark on that journey by rhetorically asking if you're curious about learning more about this. I hope that I've been able to pique your interest about some of these topics. And the reason is because one of the genesis to the reasons I wanted to talk about this is after I think we visited last time, we talked about non-conscious memory, we were having a very delicious rum drink over at Prickly Bay. And we had a nice conversation. I don't know if Randy remembers it or not, but there was actually some things in the news at the time about the role that curiosity plays in memory. And so we actually started talking about that, and I promised that if I came back to Grenada, that we would talk a little bit more about it because it actually related quite closely to some of the work that we were doing. So at the time, about this time last year, there was a lot in the news about curiosity boosting your memory. We saw basically some new studies come out, and it kind of blew up in the media. It was a very, very viral story. So Scientific American was saying curiosity prepares the brain for better learning. Guardian was saying it improves memory by tapping into the brain's reward system, is covered in the Huffington Post. So memory curiosity is its own reward. Not so much for the cat, as you might imagine. But so this was basically representing. Yeah, you got the cat. Okay. So this is representing some really great string of studies done by some colleagues of mine from my graduate lab, namely Matthias Gruber and Charm Ranganathan. And so I'll share with you a little bit about what they're finding and then follow up with a little bit of some of the studies that we've done, trying to understand some of the related mechanisms involved in that. So what Matthias Gruber had done together with Charm Ranganathan's lab that had come out just recently this past year was they wanted to study the role of these brain states that I've alluded to, and I'll get into more detail as we move along, but particularly in the context of things that might make us curious. Okay, so what they had done is they used fMRI to study human memory. So while people were in an fMRI scanner, they studied a series of things first, and they were asked to basically rate how curious you are about the answer to a handful of trivia questions. So who was the president of the US when Uncle Sam first got a beard? A bunch of really inane trivia questions, and some of them we might care about, other ones you just might not give a flying hoot about, right? Which is exactly what you're tapping into, because we want to see the variance along that sort of spectrum, and people rated on a confidence scale how curious they were. And then you get another one, what does the term dinosaur actually mean, yada yada yada. And what they were able to find is during this period of scanning, there was a couple areas of the brain that exhibited preferential activity patterns, particularly for the times that people were very curious about this information. So you can think of this period of memory as basically being the learning phase. And in fact, it's actually a time period before they're actually learning additional information. I'll expand upon that in just a second. But we found areas basically related to reward, the nucleus accumbens and the substantia nigra ventral tegmental area. And what they found was that they both exhibited this sort of linear monotonic relationship, particularly up here, and then of course the specifically higher curiosity rating related to the substantia nigra VTA. Areas very involved in the dopaminergic reward system that were specifically activated when people were interested, that they just happened to be more curious about this. And I gotta remember, these are normal undergraduate college students, which means they're pretty much zoning out for 10 bucks an hour in a scanner that's, you know, making all these crazy noises going around, and they're just tied up in this little board, a scanner. You're not that interested in a lot of this stuff, right? So the stuff that they were particularly interested in you know, this normal contextual experimental setting, really did a lot to engage in this action in the system. Okay? And what they did after that was then they actually gave the real learning phase of the experiment. So now they actually studied some information. So again, you had these trivia questions, okay? Now you also had the answers that went along with them. So what does the term dinosaur actually mean? Well, it turns out it means terrible lizard. And there are some interweaving time periods. And what they did was they were very creative about this, is they used this explicit memory. So basically they know they're going to be tested for their memory for these trivia facts. And in between, they had different faces. This is actually the study's lead author, Matthias Gruber, and they interleaved a handful of different random faces during this presentation. And the students who were taking these tests, essentially, didn't know that they would later be tested on memory for the faces too. So what it allowed them to do is gain leverage on both explicit memory for the facts and the trivia questions, but also incidental learning. Things that they weren't actively trying to encode or remember, and tap into that aspect as well. And as you might imagine where this is going, they were tested on both aspects. We'll take a look at what they found along with that. So when you look at the actual brain activity during this real learning phase, as opposed to the preceding phase, they found a couple of really interesting things. So first of all, if you just look at the behavioral measures, so in this case, this is just percent recall of how well they remembered what the answers to those trivia questions were. It turns out that in the instances in which they rated they were highly curious, they actually remembered stuff better. So at a purely behavioral level, it seems that curiosity is really playing a key role here, which might not be so surprising if you kind of reflect for a moment on those really awful classes that were dreary to take, because you just didn't give a hoot about what they were covering, but you had to do it for a gen ed or whatever you had to do. And then there's those times when it might not even be for a class, and you just find yourself engrossed on some internet blog with some topic you're really interested in, and you happen to remember that stuff a little bit better than what you're trying so hard to cram into your head for classes that you just know you're never going to use calculus again or something like that. Okay, so right at the behavioral level, we have some really sort of useful findings that curiosity does indeed seem to matter. Okay, and then what they did is they went back and they categorized the brain activity during these different time periods, and you can identify times when they answered that trivia question correctly versus the times that it was forgotten. So this is that explicit memory measurements, and they're actually measuring activity during that trivia question. What they found was that areas that were related to both reward processing and memory seem to exhibit greater fMRI activity, namely these areas in hippocampus. That's an area that's famously involved with a lot of memory processing, both encoding, consolidation, and retrieval, as well as this dopaminergic reward network, substantia nigra, VTA, and nucleus accumbens in the midbrain. So what that's basically showing is that the highly curious condition exhibited a lot more activity than the low one for most of these areas. Okay, but if there wasn't curiosity that was reported by the subjects about a certain topic or a trivia question, those areas didn't seem to exhibit such sensitivity to encoding that information. So starting to link some of these biological areas and their physiological activation patterns together with the behavioral increase that we saw at first pass. So when we look a little bit closer for that, now we can also ask questions about what might be involved in supporting that incidental learning that I introduced a few moments ago about happening to remember the faces that we're in. Now we can recategorize the same pattern of brain activity during that trivia question based on whether or not they later remember the face or if they happen to have forgotten it. So what this is actually tapping into, remember this is a little bit later down here, not during the actual question. So it's actually showing a temporal aspect to the memory. What I mean by that is, if you're curious about something and you're studying that question, will that sort of elevated pattern of activity that we just identified, will it carry over in time? A few more seconds later, and help you basically tag and pull up some other information that can also be better encoded then, on accident, right? Which is a really creative way to do it if you think about it. They're really, really smart about how they designed the study, and that's exactly what they found. So basically, if you look at the substantia nigra and the hippocampal area, both again these memory-related activities and the dopaminergic midbrain reward networks, the level of individual variability that was in this curiosity-based memory effect was actually supported by that anticipatory activity at the individual differences level. So both of them were particularly influenced there, and as well by functional connectivity between those regions. So basically, the greater memory effect there was in each of these areas, the greater the curiosity was, or the memory benefit. So it was really helped to sort of solidify that finding in that particular regard. Just to note, behaviorally, again, both the explicit memory and the implicit memory exhibited increased behavioral performance. And these midbrain sort of dopaminergic reward systems were heavily involved. So this is just a cartoon schematic sort of remind you what some of these networks are. So you have the hippocampus and the medial temporal lobe. It has a lot of bidirectional connections with these dopaminergic systems, both in the midbrain here in the VTA, as well as the nucleus accumbens. Particularly, we're looking at the dopaminergic connections in this particular regard, going bidirectionally between them there. So these instances or these areas were highly involved in reward memory, and the idea that they suggested from these findings is that the VTA, or the ventral tegmental area, basically seems to be involved in signaling the salience of the information that's coming up, and the nucleus accumbens plays a slightly different but coupled role in basically integrating the information about that salience that the VTA is indicating, together with the sort of goal and motivational related information that seems to be supported by more frontal cortices. And of course, what those connections are going to do is prime that hippocampus to bind this information together as the information is coming in. So the hippocampus is optimized to bind different streams of information together, and it seems like one of the ways that you can prime it is through this dopaminergic activity through these networks of associated connections. So it seems that basically curiosity might be its own type of reward at an intellectual level. Or it might, said it a different way, it might be that when we're curious, we're actually optimizing the brain's reward system to essentially what you might think is get excited about something that will soon be an intellectual reward in just a moment or two if you can find out the answer to this question, right? Which I think anecdotal, at least for me, kind of maps into, I think, our experiences with our own learning and memory. Again, think about really boring classes versus something that's really exciting that we learn really easy that might not even be in a class. And you're like, geez, why can't I learn my classes that quickly and that seamlessly? And so what we want to do, and what I would like to share, is some follow-up questions, which is like, how and why does this type of activity occur? And what are the brain systems that seem to underlie it? And so I'll try to share some studies that we've done that seem to then build upon these basic findings and understand some of the cognitive mechanisms involved, some of the neural mechanisms involved, and gain greater insight into what it is that might be happening. Okay? And so in embarking on that journey, it kind of requires us to reconsider some of our traditional paradigms. As I noted before, stimulus-response relationships has formed the backbone of how much of cognition and neuroscience has both approached the respective fields and the convergence of the two thereof. So you might remember some of the famous Pavlovian conditioning. You ring a bell, the dog starts to drool in anticipation of some food. You have this fundamental change from unconditioned responses to conditioned responses. And we know that the hippocampus plays a key role in binding these different types of associative information together. And it was not just Pavlovian conditioning. We know operant conditioning, Thorndikean conditioning, things like that have been really prominent backbones of the field, but we also know that historically behaviorism, at its essence, has fallen short, namely by virtue of the cognitive revolution that came about, spearheaded by Earl Miller in the 50s, which said behaviorism, these basic stimulus-response relationships cannot be sufficient in their own right because there's other contextual information that's setting the stage for that beforehand that really changes how our cognition treats these basic stimuli to create a response. Now, the cognitive revolution is very famous for contributing this aspect to moving beyond pure behaviorism or classical and operant conditioning. But historical and modern science, particularly our neuroscience and our neurobiology, has completely ignored that for the most part. And here's what I mean by that. We still model neural activity as a function of stimulus onset. And what I'll suggest to you with a couple of studies that I'll present in a few moments is that might be a nice way to do it, but perhaps an incomplete way to do it. And so there might be more than just black box of responsiveness. A stimulus comes in and it produces a response that ultimately supports our thoughts, behaviors, actions beyond our mere response to stimuli in the environment. So the main method that I'll present today is electrophysiology, so namely EEG and some ERPs, which are average electrical potentials. So when we record EEG from the scalp, it's a very nice way to gain high fidelity, high quality, extremely precise temporal resolution about the information and the type of physiology that's happening during cognition. It's not so good at spatial resolution, but if your questions are about when things are happening and how, it's a fantastic method. And so you can record from the scalp, you can see they oscillate in different frequencies, and we'll plot that on some data that I'll show that look like these spectrograms. So what you're doing here is you can plot frequency on this y-axis, and time as we move along. This vertical line here represents that stimulus onset, canonically, as we've talked about a little bit. And so you can look at activity before the stimulus, you can look at it after, and you can see differences shown on color maps, basically higher activity, lower activity, and just like we can in traditional neuroscience methods, fMRI, EEG, ERPs like this, you can subtract those conditions and see differences represented by these color maps. So we'll talk about some data that look like this. I'll also show some EEG and some ERPs, which is the average of that over hundreds of trials. I'll talk a little bit about some nerve imaging as well, some fMRI patterns, but notably, the time lag on that is a little bit slower, 2 to 4 seconds versus an order of milliseconds. Okay, so we'll compare and contrast those two, and they offer complementary approaches. And particularly, we'll talk about some of these studies using human approaches to cognition. And we'll see that this type of activity can also be mapped topographically on the head. So when I show figures like this, you can imagine like a bird's eye view looking down. So there's a nose, a left ear, a right ear. And these different color maps are basically topography maps. So if any of you guys are hikers and you're used to topography maps, or you're sailors and you're used to underwater topography maps and things like that, oceanography, the idea is the same. These contours and colors represent basically peaks and valleys of magnitudes of activation, of where that peak is, for example. So just a little introduction to the type of data that I'll be talking a little bit about. We introduced this idea of classical conditioning. The traditional aspects of neurobiology is focused on these stimulus-driven responses. We've seen that going all the way back to Descartes in 1677. You have a stimulus and the output, stimulus from the outside, it comes in, right? We're just a black box responsive unit. It produces our behavioral output, right? So you have a stimulus, it leads to a response. And we can see that traditionally modern neurosciences model our neural activity as a function of a stimulus onset. So stimulus comes on, and things happen a little bit later in time as a function of that stimulus. And so it turns out, though, that more recent studies have begun to sort of crack this traditional dogma, or at least crack the veneer on the outside of it. And a lot of this has come from really great studies that have been done in the fields of sensory perception and attention, okay? Notably some studies by Steve Block, Ali Mazaheri, Jesse Benson has really done some fantastic work that have identified that aspects of perception and attention can suggest that the neural states that are happening just before that stimulus pops up on the screen can actually play a really influential role in how that stimulus is processed and what our behavioral responses to it ultimately are. So the question that I wanted to ask is, can these same principles that we have found from some really great work that these guys have done in worlds of attention and perception, can we find those same principles evident as operating in our human memory? And we use the EEG approach as well as some fMRI studies in order to address that. Okay? So some preceding work has suggested that this might indeed be the case. Okay? So in 2006, Linott had a paper in Nature Neuroscience that used ERP data to identify that this differences in activity before a word popped up on a screen in response to a cue, so this is basically what we call pre-stimulus activity, differentiated based upon whether that information was later remembered or not. And using MEG approaches, which is magnetic encephalography, sort of a complementary approach to EEG, again, this is our sort of stimulus onset here. We're used to looking at activity here. But the differences between information that was later recalled and that was forgotten, the brain didn't exhibit sensitivity to that after the stimulus was on. But using this MEG approach, Guderian and colleagues, which is a research group operating out of NIH at the time, found that the brain actually did exhibit sensitivity to things that would later be forgotten or recalled just prior to the stimulus onset. Okay? Now this is about 200 milliseconds, so it's really, really precise. All right? To the order to which things like fMRI would be completely insensitive and unable, I should say, to even detect. Okay? And perhaps even behavioral measures. So EEGs and MEG here is really giving some nice insight. Now there had been some initial work from fMRI as well, identifying some of these dopaminergic reward systems and some hippocampal activity. And I'll talk about that with some follow-up studies in just a minute. But when we talk about the EEG, what we did is we wanted to ask, well, that's really nice and really interesting about the learning stage, encoding of memory. But don't forget, after it's encoded, it then has to be consolidated. And then, presumably, most people here are teachers or professors in one capacity or another, or you will be very shortly. And what we're really interested in is we need people to retrieve that information. Once it's learned, once it's consolidated, we have to give people tests because we want to know, can we certify you with a degree that says, yeah, they kind of know this stuff, right? So what we want to know is can we find the same principle evident in retrieval as opposed to the learning phase? And so we recorded EEG. This is my lab at UT Dallas. We're recording these electrodes on a healthy, normal undergraduate human. And we collected this during 200 words that they studied and particularly measured EEG during a memory test. And what we were able to do was, instead of looking at the EEG activity during the stimulus onset, which is what traditional modern cognitive neuroscience has typically done, we looked at the activity that was occurring for about a second or so beforehand. And what we were able to do is categorize that brain activity based upon how they responded to what we'll call an item memory task and a source memory task. So for the purposes of conversation today, just know that this allows us to tap into that episodic memory. So it's not just knowing what Juno likes to do, but do you remember the pictures of her doing it, that rich contextual episodic tapestry, okay? And we categorized that activity based upon those responses, and we looked at the upcoming brain activity to see if it was related during memory retrieval, okay? And so what we found is when we record this EEG activity, we can look at these topography maps. What we're looking at here is a difference or a subtraction between the times that people remembered literally retrieval, memory retrieval, successful episodic memory versus not, okay? And particularly source memory. And this is not your traditional activity map, but this is a statistical map. So anywhere where there's red, it's actually representing a reliable t-value. Okay? What we can see is this mid-frontal, left temporal parietal area shows this big pattern of activity right before a stimulus comes on the screen. So this is negative 150 milliseconds to 0 for stimulus onset. So what you can think of is kind of like an old Western movie, right? You got the swinging doors of the saloon. You don't know who's coming into those doors. Somebody's going to roll in through it. Right beforehand is going to determine whether or not you recognize that person from, I don't know, drinking at that saloon yesterday or whatnot. So the idea here is that something's happening right before you have to remember. Not learn, but remember. And it's predictive of how they respond. So when we look at this midfrontal slide, we can look at the spectrogram. This is stimulus onset. We can see that activity is happening in the theta frequency, about 4 to 8 hertz And this is really the big piece of action here where this pattern of activity was significant. Okay, and it's representative of the rest of these sites. And what was really interesting is individual variability here also predicted behavioral responses. Okay, so we know that we can see a group difference in these maps here between our memory performance, but when you actually measure the magnitude of how big this difference is in pre-stimulus data, it actually predicted the magnitude of their behavioral performance on that task. So it was actually tangibly related to behavior, and it seems to predict our accurate memory retrieval, not just the ability to encode. And so to follow that up, we wanted to next ask the complementary question. Well, this is really nice physiologically, and the really, really temporally precise information of the physiology, but what are some of the neural structures involved that EEG is absolutely horrible at identifying? And so for that, we returned back to an fMRI study and particularly used that, the learning phase to ask this question. Particularly if you remember a few slides ago, our learning phase pre-stimulus activity was much longer in time. Remember those two ERP traces? So because it's longer in time, it allows fMRI to pick it up a little bit easier because it's so temporally insensitive. So what we did here, actually, let me tell you one more quick piece of information about this, because it maybe can help to answer some of the questions about the network dynamics that can help motivate some of those neural structures. And what we did is we correlated this pre-stimulus activity with actual post-stimulus activity. So people still remember, and there's still memory happening, so there's still neural activity that's supporting the actual retrieval, and that's pretty normal. We see this like parietal activity. So we wanted to correlate our frontal, midfrontal parietal theta beforehand with our post-stimulus activity, and this is kind of what this is showing. So where these arrows start, I know this is kind of a wacky graph to sort of digest if you're not used to looking at it. Where the arrows start represent our pre-stimulus activity patterns for the theta frequency, and where they point to represents where they're related to post-stimulus activity. Okay? And now the colors represent the statistical magnitude of that relationship. So you have a lot of information shown here, and what this is showing is that pre-stimulus activity, okay, about negative 150 milliseconds, is highly related to the amount of post-stimulus activity that's there, about 300 to 600 milliseconds normally after the stimulus onset. So what it does here, what this does, it gives us a really nice bridge to say, we find this really new, interesting thing, but it's not completely wacky because it's actually related to the really fundamental traditional effects that we're used to finding post-stimulus. And so some control analyses are here just showing that temporally, it's not happening immediately after stimulus onset, it's not happening later, but it's a very temporally precise effect that corresponds to these findings that we saw here. And so the idea is that this frontal theta might be setting the stage to start maybe prime our computational neural networks to allow us to engage in that pattern completion process that many people think seems to be the computational support of our ability to engage in set-aside retrieval. So that being said, short diversion there to understand some of these network dynamics. We want to look more in the spatial domain, and I'll return back to talking about that fMRI study. So what we did here is we now wanted to know a little bit more about associative memory. So remember, we're binding two pieces of information. So people studied either a pair of pictures, or excuse me, a pair of pictures, or a pair of words. And whether or not they were having pictures on the screen or words on the screen was informed to them beforehand based on whether they got an X or an O. So people, if you saw an X, it meant you're getting a set of pictures coming up. If you got an O, that meant you're getting a set of words coming up. So first, gave them a little bit of a cue, and we used fMRI to study this learning phase, and particularly wanted to look at this pre-stimulus area prior to the onset of the stimulus, and of course we categorized the brain activity based on their later responses at retrieval. Okay? At first pass, what we found is this broad level of cortical activity, where there was higher activity patterns for times when they had a successful associative memory, remembering the two pieces of information that were bound together, as opposed to times when they had a failure, or what we'll call essentially a false alarm in associative memory, which is remembering falsely thinking that information was paired together when in fact it wasn't. Okay? We can see it at frontal parietal cortices, and then more medially the posterior cingulate and parietal cortex as well. And we can see that temporally it's happening really, really early without stimulus onset, which is actually incredibly precise given the imprecision traditionally of temporal measures of fMRI. So actually took a really long time to do this study because it was pretty tough to get that temporal resolution dialed in. But we also found that not just in the cortical areas, but subcortically, we saw the same pattern of pre-stimulus effect here in the hippocampus, and we could reliably dissociate that from post-stimulus activity in the hippocampus as well. So we can dissociate pre- and post-stimulus activity, and we can find these patterns of activity evident both in the cortical domain as well as in the hippocampus. Okay? And so what we wanted to do is ask, what are the cognitive mechanisms that seem to actually be influenced by this pattern of neurobiological activity? Right? So we've talked a lot about these neurobiological activities. We've talked a little bit about how curiosity seems to be inducing it. The real question is sort of like, well, at least for me, right, I want to know about cognition too. Like, well, what's happening? Why? What are the mechanisms? And so what we did was we correlated this patterns of activity with our behavioral responses as well. And I'll walk through this in a stepwise fashion. Okay, so bear with me. We're going to try to break it down. We took these pattern of cortical effects as well as our hippocampus effect. And first what we did was we ran a multiple regression analysis, basically a correlation of each of these patterns of activation neurobiologically with the behavioral measurement of how well they did on the memory test. And what that behavioral measurement was is basically the subtraction of the probability of the successful associative memory hit minus the probability of a false alarm. It's a pretty standard measure for how do people perform. And what we found was that each of these patterns of activity was actually negatively related to performance, which is like, oh man, this added like a whole nother year of analyses to figure out what the heck is going on, right? But not only was they negatively related, okay, but in the multiple regression analysis, it turns out that each of these effects, cortically and subcortically, accounted for statistically significant different amounts of variance in the behavioral data. So they both predicted a negative relationship, but it was a different negative relationship. So then we started just banging our heads against the wall because we're like, geez, how do we make sense of this? So let me share with you exactly how we tried to do that. And it actually became a really elegant story. And so what we were able to do is I said, well, everybody always traditionally compares these brain measures to these behavioral measures, which are actually two different behavioral measures. What if we break this single metric down into their constituent parts and see how the brain activity relates to them? So we'll break it down based on hits and false alarms, and that's exactly what we did. When we first broke it down on the probabilities of making a hit, we now see that these two similar patterns of activity now differ. Okay? So there's no relationship here for the cortical effect, but wow, there's actually 0.59 in the negative direction relationship, of course it was reliable, with the probability of making a hit in the hippocampus. Now remember, like I said, that they accounted for different variants here. Well, it turns out when you look at the probability of making a false alarm, we understand why there's differential variance in these two initial metrics, because whereas the hippocampus accounted for less likely probabilities of making a hit, it was the cortex that showed a really strong, reliable relationship to the probability of making a false memory. No wonder they're both showing negative relationships to behavior, but we now understand the cognitive reasons why. They're doing really different things, right? Which is really cool and surprising, but also at the same time, this is the cortex and the hippocampus. Of course they're doing different things. They should be doing different things. Now we can just understand why a little bit more. Okay? So of course the hippocampus was completely insensitive to that measure as well. And so the idea here was that the cortex was basically exhibiting that. It was not the same effect as the hippocampus. The hippocampus was related to the hits, and the cortical effects were related to false alarms. Okay, so let me see if I can break this down in a little bit more of a sort of a synthesized message about what this might mean. So we have our cortical effects, we have our hippocampal effects. Okay, and cortical effects are related to probability of false alarm, as well as the hits being related to our hippocampal effects. Now, it seems that the more correct memory that people had, okay, you had higher activity, but yet the more that people had that higher, amount of activity, right? Higher activity per hit. But the greater the activity that you had actually meant that you were more likely to do worse overall. Okay? Now, if Prashant should ask me, are there questions about this? If I need to unpack it more?
It doesn't make sense.
It doesn't yet.
It must.
Yes, and I will humbly try to suggest to you a way that we think that it does. So the idea is that these cortical activity patterns preceding seem to be related to false memories, hippocampus, due to lesser accurate ones. And here's the idea. So with the false memories, the associative false alarms in the cortex, what are some reasons that that might happen? Well, one reason is that you might see one of the items in the pair. You might go, because remember, these are just undergrads bored out of their minds in an fMRI scan. And they're like, well, I don't remember this pair. Oh, but I do remember that one part of the pair. So I probably saw the pair before. Right? So a single item can seem very familiar, leading us to falsely guess that we saw that pair before. Okay? So an example of that might be here. Okay? An associative false alarm. Here we have Donald Trump and Ronald Reagan. Right? Very easy to assume that these pieces of information were previously paired together. In fact, we see them paired together here. But were they paired together in the past? Anybody know? They're both leading Republican candidates for president, right? So it's very easy to assume that these two gentlemen have an associative pairing. But in fact, that would be an example of these associative false alarms. Because, in fact, Ronald Reagan was never supported by Donald Trump. In 1980, he supported Jimmy Carter. In 84, he was a big supporter of Walter Mondale. Not what we would naturally think, right? But we would have this associative false alarm tendency in that case. And so that's kind of what we think is happening here. So the more that you remember a single item, but not the successful pair, is going to lead you to make these false alarms, and it's going to be associated with cortical activity. Okay? So basically, we're remembering the wrong pair because you recall one of the right individuals. Now, let's see if we can dial this into a broader framework of how this is going to be operating. So it seems that these hippocampal effects are obviously distinct from the cortical ones, and there's this negative relationship between the cortex and the hippocampal effects, which seems that something is deleterious to memory. Okay? So, we talked a little bit about this idea of the false alarms and cortical effects. So if you're encoding single items instead of associative binding, right, we can lead to these false alarms by virtue of this familiarity process of leading to us to sort of jump to conclusions, like in the Office Space movie. There's the jump to conclusions mat, right? Because something seems familiar, we jump to that conclusion. Oh yeah, they must be paired together. And we completely forget that Donald Trump was a Democrat for 40 years. So we can jump to those conclusions. They don't always have to be accurate. A lot of times they are. So the idea here is, how can that happen? Well, it might be that there could be additional cognitive load happening during this pre-stimulus period. I'll talk about that in a minute. That might be leading to subjects being distracted. Okay. Now the other thing we talked about was these hippocampal effects. And the idea that we're kind of operating with this is this notion of resource depletion. So either our pre-stimulus activity can deplete our encoding resources that are no longer available at the same level a few seconds later when you're actually forming those associative bindings. Or they can also reflect engagement of basically what we call retrieval resources. So retrieving the instructions associated with the cue type. So during this pre-stimulus period, remember you saw that X, you saw that O, and it said it's going to be a picture or it's going to be a word. Well, how do you remember that? Well, memory. You have to remember that episode in the past, the instructions of what X and O means. So you might see the hippocampus active ahead of time because it's remembering that relationship bound together of what the X means and what the O means. And that might deplete the resources available in the hippocampus perhaps a second or two later. It's an operating hypothesis. And I'll try to show a few ways that we've addressed testing that hypothesis, but at this point the idea seems to be that there's still these brain states we can identify just before we learn, which seems to predict both if and how information is retrieved. So we talked about curiosity plays an important role, these brain states are important. Well, now we're gaining more leverage on it because we're understanding more of a mechanistic idea of the how, right? These false alarms, these hits, and they're broken down in reliably dissociable ways. Okay, that's what we talked about here. So, let's talk a little bit about these X's and O's. Okay, I mentioned that we have a hypothesis that I'd like to submit to you. Okay, so this is our task. We have a cue that came on the screen, an X or an O, and it told them whether it be pictures or words coming up later, and then we measured brain activity immediately before the stimulus popped up on the screen. Okay? So when this cue comes on the screen, it requires memory being activated to remember what that cue means, perhaps activating a hippocampal retrieval signal. Even though we're talking about memory encoding, we still have to retrieve something about what the instructions mean. At the same time, that might orient our cognition towards the material type of the stimuli, right? Is it going to be pictures or words? So we start thinking about that, right? We start expecting pictures, we start expecting words. What's the problem with that? Any ideas?
It pulls resources away from the task.
It does exactly that. It pulls resources away from the task, and particularly, an even bigger problem is that knowing whether it's pictures or words is completely irrelevant to later remembering it. It turns out it was like basically a bad part of our design on accident, right? But it allowed us to gain insight into what's happening because it turns out we never asked them about whether it was pictures or words again, and it had no bearing on the ability to encode the information. Okay? So it's completely irrelevant to successfully encoding. And so some people are going to realize it's irrelevant, right? So all you guys are the smart people in the room. You guys would probably sit there in the scanner and go, all right, whatever. You don't even need to pay attention. Why? Because it's going to take resources away. Why am I going to waste my time chasing rabbits if you're hunting elephants? Everybody, you're familiar with that old maxim, right? Somebody famous said it at some point. I don't know who. Maybe I'd like to think it was Hemingway or somebody, but perhaps not. He said, if you're hunting elephants, don't waste your time chasing rabbits along the way. Stay focused on the task at hand. Okay? So some people are going to realize that this is irrelevant, and they're not going to focus on it. But other people will not realize that, and they're going to be distracted by this. Increased resources, less ability to bind the information together, more probability of making a false memory, because you do remember one piece of the information, but you didn't successfully have all the resources to bind it together. Make sense? So it made no sense at first, but I did promise that I would try to make sense of it. And that's kind of the idea. People who are distracted by this cue information will need to draw more upon cognitive control resources. Now, for those who are less familiar with the cognitive neuroscience literature on cognitive control, it turns out that when people are engaging cognitive control, it very famously activates a frontal parietal control network of activity, which is incidentally exactly what we happen to find. And in fact, as you already know, reliably dissociated from our other patterns of activity as well. And so, basically that's going to lead directly to our worst memory, okay? And those worst memories are going to be reflected by the false alarms, like these, okay? And so, how can we test this hypothesis? Well, what we did is we ran a follow-up study. We did the exact same experiment. We showed the X's and O's, but this time they were completely random and had no predictive value about whether it be pictures or words coming up. So we took away what I wager, together with Randy, it sounds like as well, is a really distracting thing. We put in there an accident at first, but it turns out when we look at that, we'll do the exact same experiment. Okay, now it's not cued to pictures or words, and I'll show some basic patterns of activity from those same regions: posterior cingulate, the precuneus, right temporal gyrus. What we see in our very first study. This is not new data. This is what I just showed you. They're just bar graphs, but it's going to be useful for comparison purposes. All right? Information that we remembered is always higher than that which was forgotten. That's the pattern we just looked at, right? But when we took away that informative cue, okay, that pattern now reverses. Gray is now less than orange in each of these areas, okay? So now, it's always less than our rearranged pairs. That is, the activity for correct
Okay, so we've not just changed the pattern, we've actually reversed the direction of it. Okay, so it's not just gone, it's actually reversed. Okay, and what we can do is we can basically look at this and quantify it with a simple ANOVA test. And what we'll do is compare the experiments to each other as a function of that. So that's what we're looking at here. Anywhere in green is where those experiments differ in the memory effect, the difference between those gray and orange bars. Okay, this is our experiment 1 when we use an informative cue. We see this positive difference between memory, and here we saw that negative difference that we just saw on those individual bar graphs. And it's evidence all across the cortex in the exact same spots that we saw before. These frontal parietal control networks seem to be indicative of cognitive control, perhaps moderating the relative amounts of distraction at the individual difference level. And so it seems that these are deleterious effects and that they're reflecting that cognitive control. So basically, the more distracted that you are, the more you will need to use cognitive control. The higher these activities networks are going to be, and if you need these activity networks, because you are distracted, if you're in Vegas, you should bet that those people will probably perform worse on the memory test. Right? Even though, so greater activity doesn't always mean better stuff. Right? It's kind of like when credit rating companies give you a worse credit score because you've had too many credit inquiries that year. Lots of activity doesn't always mean good things. And you get dinged on your credit score because it's like, oh, everybody's pulling your credit. Well, you're like, well, I'm trying to buy a car, buy a house. But, more is not always better. So that's kind of the idea. We saw the same thing evident here in our hippocampus as well, perhaps reflecting that resource depletion idea from retrieving the information that we talked about before. So it seems that these brain states prior to stimulus is predicting both if and how. We've seen that both in the physiology as well as the structural aspect as well. And it seems that these pre-stimulus effects can be evident both cortically and hippocampally, can be both beneficial and deleterious to memory across both of these experiments. And it seems to reflect this optimized network that we identified in the physiology, correlating electrode spatial locations with the different temporal time points of activity, and perhaps reflecting the abilities to successfully engage this pattern completion process. There's a lot of researchers, Craig Stark and Mike Yassett, talk a lot about pattern completion, this ability for the hippocampus. Yeah, exactly. The ability for our cortex and hippocampus to be able to dissociate those things. And they can reflect this cognitive control of managing the interference and divided attention. So perhaps our stimulus response models of learning memory are inadequate. And if we can expand that to a third dimension of looking at time points, not just with stimulus response, but now a third dimension, it might be able to provide a more complete account of some of the non-conscious factors that support our learning memory besides just the stimulus and response. Maybe if I can zip through 5 minutes.
Yeah.
Sure.
Fine.
I'll try to share
I'm good.
the implications.
That.
Yeah.
For the 3 people left, we're on.
Well, we're in TDS.
So, what can we do to take advantage of this? Can we manipulate the brain? Okay, can we improve performance? All right, and so I'll share a little bit what we know from some people's studies that have been done. So, Matthias Gruber, the gentleman I mentioned earlier, we did the curiosity study. What led him to that study was some earlier work he did that found that these pre-stimulus effects, here before a word pops up on the screen, okay, can actually be under cognitive control. So people asked when we first reported some of these findings, we always had people ask like, well, can you really like zap the brain or take a pill or do something? How do we get this? And I'm like, well, you don't have to do any of that. We can see these patterns of activity are actually under voluntary cognitive control, which is really useful and it's a whole lot cheaper, right? So we can find that. We can also see that if you can track brain activity real time using fMRI, so this is work done by John Gabrielli's lab at MIT. In 2012, they reported that if you measure different patterns of fMRI activity, they can identify what they basically labeled good brain states and bad brain states. Okay, so basically times we have like what they call a reference or a control condition brain state. normal brain activity in blue. Anytime where it differs from that, right, you can identify as either good or bad based on, well, let's present stimuli on the screen right now, because we know that now the brain could be in an optimal state to go to perhaps a deleterious one. Well, turns out that's exactly what they found. Memory improved if you presented stimulus during a good state of activity differences versus when you presented that stimulus to learn on what they call a less good state or a bad brain state. Sort of an oversimplification there. Forget about the names. We can go Shakespearean about a rose by any other name. But the idea is that if you present using real-time tracking of brain activity, you can change performance, it seems. Now, you can also look at this in different clinical populations, and this is what Michael Cohen did during his graduate work at UCLA. He's now a postdoc at Northwestern, and he looked at different pre-stimulus brain states in normal aging. So it turns out that in aging, these patterns of activity in our older age subjects completely attenuate, whereas they're very, very much evident in our younger groups. Interestingly, it didn't change behavior because it seems that our more aged subjects were using compensatory strategies to improve their behavior. Dare I say, they might have been more efficient and garnered some extra skills in there.
Old age and treachery.
Well, they learn how to do things better, perhaps. Perhaps they don't need that. That's still an open question as to explaining and accounting for those effects, but the idea to suggest is simply that it can vary in different clinical populations. So it's really an interesting line of work right for research. And what we followed up our initial pre-stimulus theta idea with was a study that's really been championed by Brooke Roberts, who was a postdoc right when I was leaving that laboratory. She picked this study up that we had just finished doing. And what she said, well, what if we stimulate the brain at different time points? So she did the exact same paradigm. We used all my code and scripts that I wrote and everything. She did the exact same thing, except this time she did what's called brain entrainment. Okay, so she gave audio and visual stimuli at exactly 5.5 hertz, which is exactly theta. Okay, now as you imagine, you split this up into control groups, and people who got that stimuli, people who didn't. Then you ran a second experiment, those who got this 5.5 hertz treatment, and then we did it at a different alpha frequency, 14 hertz, as another control condition. So first you have a control of nothing and theta frequency, then you have a second control of theta and a different stimulation to account for both possibilities there. And so behaviorally, we found that both of those experiments saw increases in basic behavior when they had the theta entrainment. Audiovisual stimuli, real simple commercial software system, and this is during the consolidation phase, right before memory retrieval. Now, we can see that memory was enhanced, and the idea was then to look at the brain activity, and what we can see is first of all, see a bump in theta. So this is a spectral graph. It shows our different frequency components, okay? That in red is our 5 hertz versus theta versus our 14 hertz frequency there. So what we're seeing here is simply that the manipulation worked. You stimulate it at theta, and you measure brain activity, and it shows a bump in theta. If you stimulated 14 hertz, we see a bump here at 14 hertz. So first, that didn't have to happen, by the way. So it's a really important control piece of data to show that we are successfully achieving a mapping of stimulation to brain activity, right? So the brain doesn't have to care about the rate at which it's stimulated, but it turns out that it does, and we can see the topography map of where a lot of that's happening. Seems to be these left temporal parietal areas that we saw in our earlier data as well. Now, the question here was it related to memory? And it turns out, as you can imagine, I wouldn't be talking about it if it wasn't. And we can see where a lot of those activity effects were. Remember, this is the exact same paradigm, exactly the same paradigm that we did in the 2011 study. And remember that was mid-frontal left temporal parietal, mid-frontal left temporal parietal, which is pretty, I loved it when I saw it. I was like, thank you so much, Brooke, this is so cool. And she's really done a fantastic job on it.
And when you look at that, and you subtract at the group levels, the brain activity patterns for the groups that got this from stimulation and those that didn't, that's what we're seeing in subtraction here. You can see this big stretch of theta all the way across the time period in the group that actually got that theta stimulation. And it was actually related to that greater theta during the actual increased memory performance. So you can see behavior was increased, the physiology was increased, they were related. It was pretty wild when you think about it. It was a really, really exciting study. So that's currently under submission. It seems that we can actually train our physiology beforehand in order to support these patterns of activity when you actually need it during the actual memory process. So we were really excited about that. And so I know that I've probably gone over a little bit of time, but I really appreciate your patience. But that's kind of the idea here. We can help memory with different brain states, and it can also hinder memory, so these brain states matter. We know that curiosity and motivation are really important to improving memory, but also this physiological training can as well, because it seems that memory can be improved during our proper brain states, and this training can also help to induce it in the same way that our cognitive control can, or by simply inducing that curiosity as well. So the idea that we're kind of working forward with is, can we see more of these things in clinical amnesia groups, some of these network level dysfunctions? Perhaps that might be something underlying why there might be amnestic impairments. Perhaps there's disruptions in these ongoing networks, brain states, and can we use some of this theta training, for example, as a target for an effective strategy to improve some of these deficits, not just in clinical patients, but also in our normative populations as well. I'm sure we all like to forget less stuff. You know, I don't know if it's quite there yet for full application, but it's ripe for some experimental study, and perhaps we can begin to characterize some of these deficits, schizophrenia, depression, TBI, based on disruptions to these dopaminergic mesolimbic reward systems related to curiosity. So I'll skip over the last stuff that we can talk about later if you're actually just interested. These are the strategies, but acknowledge a lot of the collaborators: Andy Linus, Andy Linus, Tom Ranganith, Arna Ekstrom. Bert Roberts just did an awesome job with that theta stimulation thing. It's so cool. Andrew Wachtros was a co-first author on the first theta study that we had. And then we've gotten a lot of great funding from NIMH support, Marine Biological Institute, University of Chicago helped support some of that last work I was talking about, as well as the EC Data Center for Neuroscience and the Nursing Program of Neuroscience supported a lot of the early work that I was doing as well. So I have to gratefully acknowledge their contribution, but also gratefully acknowledge not just Juno, but your guys' time, patience, and attention this afternoon. Like I said,
would you mind just showing the slide with you had some tips for, yeah.
Sure, I'd be happy to. So,
yeah,
let's see here. Okay, so the idea here is that, just like you mentioned before, we can have pattern completion or pattern separation processes that are served by physiological signals, both in the hippocampus and cortex, and we know that all these neurons form connections that support our memory.
And they shift with age.
And they do shift with age. And basically the idea is that the hippocampus is serving this role to bind the information as an item together with our information of context. Again, sort of like we started off talking about, the information of like what Juno does with the context of like all the cool stuff that we like to remember about it, right? And so that seems to be the role of the hippocampus, is binding this associative information. And so those connections can be served by basically factors of context, experience, repetition, and of course our cognition. And so some of these strategies that you asked for, basically these pre-existing states matter. Curiosity, as we talked a lot about, which doesn't have to be intrinsic, right? You can fake it till you make it, right? You can pretend to be interested in something. Most of you guys have probably been doing that this afternoon for the last hour, 5 minutes or so, right? There's ways that, I remember learning really boring, awful stuff in graduate school sometimes. Some of it was really cool and interesting too. But there's times where like, we can trick our system, we can make ourselves more interested, or make ourselves more interested in it.
Right,
part of that is
over.
I'm sorry?
Right up to the exam's over.
Well, hey, whatever works, right? And so the idea in terms of if you want to, enhance our curiosity or theta, dopaminergic things, part of it is, so presumably some of you guys teach classes and whatnot, right? What sometimes I find barriers for students when I teach is they don't always understand, well, why is this information important? Right, and that's on us as instructors, right? When we start off, you want to tell them, okay, we're going to talk about blah, blah, blah today, and here's why it's important. Because it might be some weird system in the kidney or something. Well, we forget that we need our kidney when it goes, So part of that, I think, is we can tap into increasing that amount of curiosity. And so we can make some of those more boring and mundane pieces of information more interesting. So we make up an elaborate story. I had an assignment I gave to students last year to come up with neuroscience rap songs. Right? Take what they might think is really boring science information, make it interesting. Put it to the tune of a really popular song they like.
Gangster's Paradise.
Gangster's Paradise. Seriously, I mean, I have a whole YouTube channel, Comment Neuroscience, that lists all the videos that they made, neuroscience rap videos, and they had to think, well, how do I rhyme this word? I need to find another word about the brain that rhymes with this, and they really have to engage in these deeper collaborative processing. Again, they're becoming more curious in something that they're otherwise bored out of their mind about. Right? So it doesn't have to be intrinsic curiosity. Right? We can game that system. And so we know that repetition matters, right? So study, study, study. But of course, that alone, which is basic cognitive psychology that we know is not the best approach. But better than that is, of course, the depth and the meaningfulness of that learning. That matters more than repetition does. So there's a lot of cognitive psychology. A lot of this is like Cog Psych textbook 101. But elaborative encoding, depth of processing matters. And it's going to matter more than just basic repetition. And again, when you're engaging in that meaningfulness, that depth, collaborative encoding, what you're doing is you're binding more of these connections like we've talked about. It's going to help us to retrieve the actual associations rather than falsely assuming the Trump-Reagan relationship or whatever. And again, sort of some of the things that we talked about today, the role of divided attention is an old cognitive psychology story. But we saw that at play a lot in some of the brain state data that we looked at, some of the neurobiology as well. So that divided attention that we induced completely accidentally in our experiment seemed to really play a deleterious role in the brain state, not just during the learning and memory, but in the ongoing state of mind beforehand, which I think is a step beyond that old school cognitive psychology conversation, right? Don't divide attention when you're learning. Well, don't divide attention right before you're learning too. So that load matters. We talked about the role of elaborative encoding. I have this little guy here because I'm told that's the brand logo for Snapchat, which I guess is what the kids are using now to send text messages.
Kids these days.
When we're lecturing, if you see them messaging, that's probably what they're doing. Dividing their attention.
Let's be honest, it's pure Facebook, but like size.
Yeah, seriously, yes. And then the last part that I actually try to suggest to people are a couple of really good stories from cognitive psychology as well. Basically, things called the production effect and the generation effect. Basically, we remember stuff better if we generate it or if we produce it. Right? So a lot of students always ask me, can you make a review sheet? Do you give a review sheet for the test or this and that? And I say no, because I feel like I'm stealing an opportunity from them to learn, because when I make the review sheet for them, I learn this stuff a lot better. And I'm like, well, this isn't helping you. This is stealing from you the chance for you to tap, for you to get the benefits of these two really prominent effects. Those are really good aids, and more broadly, just forming some of those rich contextual bindings of those associations, because we know that's the role that the hippocampus is really playing. A lot of people are doing that, be it in pattern completion separation stories, and in all sorts of different levels, be it from the fMRI to the cellular levels. These rich associations, not just of the content, but also with the concepts, which I think brings it home to understanding why this stuff is important again, integrating these concepts with the content by virtue of getting interested and using that curiosity to tap into their performance and things like that. And so that is my little foray.
No, it's great.
It's sort of like a
It
PSA.
reinforces some of the stuff that we're doing already, but it really, the curiosity element is something that, I don't know why I brought this up with Black, we never really thought about. And it's also nice to see visually what really is happening in there.
Yeah.
The distractions and relevance and all that, yeah, it's hugely important, and why we stress so much the teaching aspect of it as well, as a way to kind of contact this. It's very interesting.
I also think the not the depth, but the contextualization. So I think, so I can even imagine, okay, well, say you're learning the Krebs cycle, if there was like a movie or some type where you could actually go in, like imagine now 3D, or like virtual reality. Imagine where this could go, right?
Yeah.
So now you put on your virtual reality that Facebook is working on, what is it? And they're supposed to come out actually with it. And you actually, now you go inside
Yeah.
the molecules and you are actually experiencing the Krebs cycle. That makes it so much more memorable because now you're getting the episodic memory that's combining with the semantic information and they're living it. They're not just trying to sit there like when my advisors come in, they're like, oh, I went through this stuff 20 times. Like, yeah, but are you just repeating it to yourself? Are you actually living it?
Yes.
Experiencing it.
Yeah,
Right?
I've had students tell me the same thing when it comes to, they all watch these YouTube videos.
Yeah.
I don't know what the names of them are, but there's like a series.
Oh yeah, there's like
Where the guy
Khan
draws.
Academy and
Yeah, he draws
Or they
them
draw
out,
the,
and they've said they can actually, on
You're
the
living it.
test, they can actually picture the guy
Yeah,
saying the thing with the drawing and everything, so yeah.
yeah. And then the curiosity too.
yeah,
You worked with Nick Rugg?
I did at my postdoc with him. Yeah.
Okay.
Do you know?
Because he's, yeah,
Yeah, he was director of the
Yeah, okay.
CL Center for Learning.
yeah, the
Memory.
Center for Neurology and Learning and Memory, yeah.
Yeah.
Okay, so I got a whole list of questions then to ask you about that study, but probably will bore these people crazy because it's really weirdly arcane, but.
Well, I'm happy to hang out
Well, anyhow.
and chat about whatever you
You're
got.
here for a week or two.
I have two quick questions.
Yeah.
The XO, the literacy effect of the X and the O's.
Yeah.
When I watch lectures, a lot of times they'll come in and maybe we'll have either flippant questions or they'll say, last time we studied this. And they're getting everybody to remember about what they said, then they're starting into a no. Your studies were in milliseconds, but we're talking about macroseconds. And that, would you say that there's a deleterious effect to sort of the encoding and the processing of new, sit there and have them focus on stuff that's not even going to be introduced and a part of the lecture by saying, here's where we were, this is what we studied yesterday, or let's do a quick question, see how many people remembered stuff from yesterday. Now we're going to shift and we're going to talk about the respiration system. I mean, can it apply on a macro level or is it only in the micro level of milliseconds?
I think it's a really good question of cognitive psychology, right? So I think a lot of us approach a lot of things with the reductionism of, say, neurobiology, and to get into the molecules, the genes, the systems, but I think a lot of the questions are actually questions of cognitive psychology. I think cognitive psychology might be a really good approach to, say, trying to answer that question. And I think that sort of what we were talking about here is, I think that if you're gonna say completely shift gears, like say we talked about this yesterday and here, I think any time that you completely shift the context is probably, I think you might risk the dangers
People are gonna fall off the truck.
Yes. Some of them were holding on for dear life. I was often that guy holding on for dear life and just praying that it hold on to you, right? But so I think anytime that we shift gears, which naturally we have to do every lecture, right? Because it's a different lecture. We have to talk about different topics. That's our job. I think we always risk losing a few people off the bandwagon, but I think that there's ways that we can mitigate that and maybe reduce some of those distraction or deleterious effects by virtue of that, by helping to make some of those bridges and connections for people between them. You know, if we know, getting them to become interested in this new thing and providing the bridge that connects the two topics. Even if it's a crazy bridge, and if there's really, it's a bridge to nowhere and the topics really are unrelated, you can still, I think, just cognitively orient them. So we talked about you know, airplanes yesterday, we're going to talk about boats today. Oh, but the link, they both do transportation. You know, whatever. I think that as educators, we can find ways to bridge that so more people don't fall off that deleterious shift. Because I think anytime we completely shift a topic or something, what we're doing is shifting the cognitive framework, the context that our nervous system is comfortable operating in at that point. And now we have to start activating different sets of neural connections that are not primed, whereas if we stuck in the same topic, that topic's primed. Those connections are going to be better suited to making those connect. You know what I'm saying? So yeah, I mean, I think that we do risk those deleterious effects at the longer scale of days as opposed to the seconds. But I mean, it's not necessarily for naught. I mean, most of you guys have been teaching far longer than I have, you know, and you guys have a host of mastery and those types of things that I think that, you know, I could probably learn from too, and how to help shift between topics, between lectures or something like that. I think. But yeah, that's one thought. I do really appreciate you guys' time and attention.